Hello and welcome to this video where I'm gonna be going over 20.1 and 20.2 in the IGCSE Biology syllabus in the new book here. And this goes into detail about biotechnology, and it goes into detail with DNA sequencing. So it especially ties into the chapters about genetics, about DNA, and it also ties into our chapter about enzymes and biological molecules. So I'm going to be going over this here in just rough details, but I really, as always, recommend reading the textbook as well. Biotechnology is the process of using a living organism to carry out processes to make substances that we want. While this might sound like super complicated and sound like stuff that's only happening in a high-tech lab, it's really stuff that we have been doing, and many of you might have done yourself. When we bake, we have a microorganism, the yeast, doing stuff we want. When you make alcohol, if you are using fermentation to preserve something, if you're making homemade yogurt, if you're making homemade cheeses, we all use microorganisms to do stuff for us. So bacteria and fungi has been used this way for a long time. But with the growing field of genetic modification, we now have way more specific ways of doing this, and this is going to change the way we see technology and biology in the years to come. So one question could be, why are we using microorganisms for biotechnology? Well, there are several reasons, but important ones are that they are small and they take up little space. If we were using elephants, bad example, but if we're using elephants for insulin production, that would take up a whole lot of space and be quite complicated. Bacteria don't require much space, neither do yeast or other single-cell organisms. They can reproduce fast, and we can use them to make complex biological molecules quite easily. There are very few ethical issues. If we're using like Labrador retrievers or elephants, many people with a good cause might say that's not very nice, but not many people have a lot of feelings about what we're doing to yeast or bacteria. And last but not least, especially when it comes to bacteria, because the way that DNA is, it's quite easy to go in and make genetic modification to the DNA of bacteria and thereby make them express the proteins that we like to have expressed. So if any of you have tried making your own bread, you've probably seen this before, yeast. And yeast, when we buy it in the supermarket, is this weird grey, somehow odd smelling mass. In reality, these are just a huge bunch of single-cell fungi, and when they are in the fridge, here kept cold, they are inactive. But when we mix them with water and a substrate, for example sugar or flour, they will start reproducing. And fungi can be used for several things. We'll later talk about how to make bread, but if the fungi here are kept under anaerobic condition, means no oxygen, then they will convert sugar, glucoses, into CO2 and ethanol, alcohol. This is the process that's being used when we make wine, beer, but it can also be used for making biofuels. And when we make biofuels, we take organic matter and then we break it down, and it's turned into ethanol, which can be used for fuel for cars. There are both good and bad things about using biofuels. The good thing is that we don't use as much fossil fuels. The bad thing is that we take up space that could be used for making food for people, and then instead making fuel for cars. When we are making bread, there are several ways of doing it, but the normal way is we take some flour and some water, and then we mix in this yeast here, and there are enzymes then that will start breaking down the long chains in the flour, and will break it down to monomers. Once it's broken down to monomers, the yeast can start consuming it. So why do we add yeast to our bread? Why do we want it to consume some of our flour? Well, it is because when it does it, it creates small bubbles of CO2 in the dough, and that is what makes the bread all light and fluffy. If you ever tried making bread without yeast, it's a quite more dense thing. So the CO2 that is created in the bread kind of make the breads we like to eat. It also makes a little alcohol, but that alcohol is broken down when the bread is baked in the oven, so it's not anything that has an influence on it. But if you leave the dough standing for days, the amount of alcohol can turn higher, and there is a hypothesis that is how the first beer was made. It was leftover from bread production that turned into beer. People tasted it and figured out, okay, this is something we'd like to continue doing. So, if you remember back at the enzyme chapter, an enzyme is a biological catalyst that speeds up a reaction. And by the name of it, it usually starts by saying what it breaks down. So pectinase is something that breaks down pectin. Now, what is pectin? Pectin is a biological molecule we find in, for example, fruit, which help the cells together, glue them together. Now, if I were to squeeze apple juice out of this apple, I of course would squeeze it and the juice comes out and I have some nice fruit juice. But it's often hard to get the last bit of fruit juice out of it because the cells, they hold together. By adding pectinase, I'll get a higher production of fruit juice. I'll get more juice from each fruit, especially when using stuff like oranges. So it makes extraction easier. It also makes the juice itself look clearer, which some people really like. We also use biotechnology in washing powders. Now, a normal washing powder or soap we use for fabric is just basically that. It's soap, and soap breaks down the fat, so it's easier to get stains out of the clothing. We can use warm water and soap, and we'll get everything nice and clean, hopefully. But we can also get biological washing powders today, which contains enzymes. And those enzymes will help break down the protein and the fat, especially if you spill like food, tomato sauce, blood, other stuff on your clothing that's hard to get off. These enzymes, they work quite well at a low temperature, like 40 to 60 degrees, but you can't use in really hot water because that will break down the enzymes, so denaturate them. Normally you say 60 is too warm, but some of them has been engineered to be able to work at that temperature range. But whenever you're using a biological washing powder, it is stated on the back specific what temperature it works with, and many of them works quite well at low temperatures, which also makes them more energy efficient. We don't have to use as warm water. Now, as they contain both proteases and lipases, if you put them on your skin, that will start breaking that down as well. And because of that, they are packed in small capsules, so they don't start breaking down your skin when you add it to your washing machine. When you're using a biological washing powder, of course, think about what fabric you're using, because if you're using something like silk or other organic materials, you might have an issue with the washing powder also weakening the fabric itself. But being able to add enzymes to washing powder is a big thing, and many Danish firms, such as Novozymes, have made a huge success on doing industrial production of these things. Another enzyme is lactase. Lactase breaks down lactoses in the milk to more simple sugars, which make them easier to digest. Now, all human babies produce this themselves, so when you get milk from your mother, you're able to digest it and get the energy from it. But as we grow older, some people, they lose this ability to produce this enzyme. I, for example, have no problem. I can drink milk and eat cheese, have no problem. But if you lack this enzyme, then larger doses of milk, yogurt, stuff like that can give you an upset stomach. So you can now get milk and cheese and yogurt where we added the lactase enzyme before selling it. And that means that the lactose is now broken down, so people who can't digest it can still enjoy yogurt and cheese and milk. So this is another example on using enzymes to help process our food in a way that makes it available to more people. Now, when we are using bacteria and yeast and other stuff to make these products for us. This is of course the kitchen scale of doing things, but when we do it in a larger scale, we often use what's called a fermenter. And a fermenter is an industrial machine where we can go in and control things. We need to be able to stir it at all times, so we don't have all, for example, if we're using yeast or bacteria, all fall to the bottom. We are also able to control the temperature. We are able to release any excess gases, and we can also add the amount of other nutrients that the bacteria and the fungi might need to grow. And this way, because of fungi and bacteria growing quite fast, we can have a quite rapid growth of our bacteria and fungi because we control the temperature, the pH, the amount of nutrients, and a very specific range that suits our organism. And that way we can have a quite high production of whatever bacteria we need. And if that bacteria or fungi is producing some biological proteins we need for further production, we can then harvest that and use later. Now, so far we've only been talking about using microorganisms who are already there or enzymes who are already there. But with modern DNA technology, we also now have the options of going in and changing the DNA of an organism. Now, just remember back to our chapter about DNA and RNA. Our central dogma is that DNA is double-stranded, and it then codes for the RNA, which is single-stranded, which goes out into ribosomes, and then we have DNA, RNA, and RNA then makes the protein. So if we go in and change the DNA code of an organism, that will make for different RNA, and that will express in a different protein, and that is what we use in genetic engineering. For example, before genetic engineering, people with diabetes 1 need insulin to survive. And before we were able to make this with microorganisms, you had to take it from, for example, dead pigs. And that was expensive, it was less safe. But when we're able to make, for example, a bacteria or yeast make insulin for us, we can make it in a large amount, we can make it cheap, and we can make it safely. And that has been a huge relief for people with a disease like diabetes to be able to have a safe supply of insulin. So the idea is that we can take a little piece of DNA and add it to a microorganism, and then it will make exactly the protein we need. Now this was insulin, but you can do this for all different kinds of protein. As long as you have the code for it and can add it to the bacteria's DNA or the fungi's DNA or pretty much anyone else's DNA, then that protein can get expressed and made. This is quite revolutionary, and it's extremely exciting. One way we're seeing this is in GMO plants, which means genetically modified organisms. And we're already now seeing that GMO plants are being used in large-scale industrial farming. In GMO plants, we go in and modify the plant, so the plant now suits our way of farming better. And in one way, this is not that different from what we're doing with selective breeding. Remember, in selective breeding, we keep looking for that one thing we want, and then we just keep breeding to express it more and more. But with GMO, we simply skip the process of the many generations of selection, and we simply go for, okay, we want this plant to express more of this protein, so we go in and add the DNA code for that. It has been used to make plants resistant to pests or to specific pesticides, herbicides, which will make farming more efficient. And it has also been used to make plants have a larger amount of specific nutrients. For example, in rice, we have what is called golden rice, where the rice produces more beta-carotene, which is a precursor for vitamin A. So children, young people who eat this rice gets more vitamin A, which is very healthy for human development. So this is just examples of what can be done. Now, many people are scared about GMO and think that we are now playing God. But in many ways, it is not that different from what has been done by selective breeding. Here we just skips the steps and moves a little faster. So if you go in and Google GMO, you probably get a lot of arguments for people either being very much for them or against them. Some of the arguments against using GMO is that we already have a wide selection of plants. We can just use other crops. We don't need to modify them genetically. And another, maybe better argument is that there are only very, very few places where these GMO seeds are made, which make them quite expensive for the farmer, and it also makes the farmer depend heavily on a very few companies who make them. Arguments for GMO is that we can then reduce the amount of chemicals we need to use for conventional farming, and that we can have a higher food production and maybe add more nutrients to the food we produce. So there are arguments both for and against using GMO, but it is one of those subjects which often become quite heated and people are very emotional about it. There is something about when we start to change the genetic code of stuff we're eating that makes people skeptic. And I'm not to say whether GMO is just good or bad. I think it, as many other tools, have both sides. It can be used great, and in some cases it might be a very bad solution. But it is interesting to see that there are different points in the argument on why you should or shouldn't use GMOs. But they are being used around the world, especially genetically modified corn and maize is a huge thing, especially in the United States. Okay, so far we only talked about we have the ability to actually go in and change the genetic code. And how is it done? Well, a lot have happened with the process since I studied biology many years ago, and it is now simpler and easier to do than when we started playing around with it in the lab. The idea is that in bacteria we have these small circular pieces of DNA called plasmid DNA, and using the right cutting enzymes, we can call a restriction enzyme, we can cut a piece of genetic code out, and then using splicing enzymes we can insert another part of the DNA. So let's say we have a bacteria here who is doing whatever bacteria is doing, and we then cut a part of it out and instead puts in a piece of DNA, maybe from humans, who make a specific hormone. Now when the plasmid is then reintroduced into the bacteria, well, then the bacteria has now the DNA code lying around. So once it starts going in and it starts splitting and multiplying, all the bacteria will now have this new piece of DNA inside it. And when they're then growing, well, then they'll start also making the protein we put in the code for. In this case, it might be like human growth hormone, because that was the part of DNA we put in there. This is, of course, a very simplified way of doing it. The process itself is more complicated than this, but in reality, you can pretty much put any protein code inside and then it will start multiplying. By the way, this is not unique for bacteria. It's just easier to do in bacteria because we have the plasmid DNA. But you can also do this in higher organisms, and it has been done on mice and on rabbits and so on. So it is also possible to go in and change the genetic code of animals as well. So this is a field that have been radically changed in the 25 years that I've been studying it, and it's really moving fast. It used to be like genetic engineering, gene splicing, CRISPR, all these things were very complex and it required a very large amount of technology and highly educated people. But it is becoming easier and faster to do new DNA technology. And like all technologies, it has the possibility to do good or bad. When humans discovered fire, it was nice, we could keep warm, we can cook food, we can keep predators at bay, but it also gives the opportunity to light other people and things on fire. So it has been that way with all inventions that humans make. It has a good side and a bad side, a possibility to help humanity get better or humanity get worse. And it is the same with DNA technology. It is not something that is just a miracle, and it's not something that is just bad. It all depends on how we use the technology. Used correctly, it can help us get a better food supply, get cheaper medicine, it can help us combat climate change and cure diseases. And used for bad, well, it can be used to make terrible diseases and biological weapons. So, like everything else, we have to kind of look at the advantages and disadvantages of the different technologies. But I think we can say pretty clear that DNA technology is going to play a huge part in your future, and it is going to be a bigger part of what we do in biology. Thank you for staying with me, and remember, any mistakes here are mine and mine alone, and does not reflect on the wonderful syllabus done by Cambridge.